In the part 1 of biological oxidation, we have seen how electrons are flowing from complex 1 to complex 4. Okay, in that process, there is a transport of hydrogen ions or protons from matrix side to intermembrane space or a cytoplasmic side of a mitochondria. Okay, then today we'll discuss about how ATP is synthesized with the flow of these protons back into the matrix. Okay, from complex 1 to complex 4, electrons are transported, and these electrons transport has caused movement of hydrogen ions, and this movement of hydrogen ions help in formation of ATP. We'll see in detail. Okay, normally flow of electrons from NADH to oxygen is an exergonic process. Okay, and ATP synthesis is an endergonic process. And this ATP synthesis is done with the help of a protein enzyme complex called as ATP synthase. Okay, this ATP synthase helps in formation of ATP. A scientist called as Peter Mitchell has proposed a theory called as chemiosmotic theory or hypothesis. What he states that electron transport and ATP synthesis both are coupled by proton gradient. At what place? Around the inner mitochondrial membrane. So this proton gradient is created around the inner mitochondrial membrane. Okay, if this is a mitochondria, okay, and we have inner mitochondrial membrane, okay, this is matrix, and this is cytoplasm side. Okay, cytoplasm or intermembrane space. Here the movement of H plus ions will takes place whenever an electron moves from complex 1, complex 3 and complex 4. Complex 2 doesn't pump hydrogen ions or protons. Okay, these hydrogen ions are pumped from matrix side to cytoplasmic side. Okay, what will happen when these hydrogen ion protons are pumped out from matrix to cytoplasmic side? Here what will happen? Here in matrix, H plus concentration decreases. More positive ions are moving out from matrix to cytoplasm. Then what will happen to the charge inside the matrix? Negativity is increased. To inside the matrix, it becomes more negative, and outside the matrix in the cytoplasm, there will be increased H plus concentration, okay, and more positivity. This is what happens with the movement of protons from matrix to cytoplasm side. Okay, now these protons which are transported from matrix to cytoplasmic side, again they have to be transported back into matrix. Okay, during this flow of protons back into the matrix will help in synthesis of ATP. So the flow of protons drives the synthesis of ATP by an enzyme ATP synthase. ATP synthase. Okay. This force, proton force, okay, is called as proton motive force. Okay, proton motive force. This proton motive force is consists of two compounds. Okay, two components. One is chemical gradient, which is caused due to movement of protons. Okay, that is change in pH. Okay, plus charge gradient. Okay, charge gradient. Both these factors will cause proton motive force. Okay, this proton motive force helps in synthesis of ATP with the help of ATP synthase. Okay, next this will see the structure of ATP synthase. Okay, ATP synthase. It resembles a ball and stick. Okay, ball and stick. This is a ball and this is a stick. Okay, this type of structure is the structure of ATP synthase. Okay, this stick part is called as F0 and the ball part is called as F1. Okay, this F0 part is present or it is embedded into inner mitochondrial membrane. Embedded into inner mitochondrial membrane. Inner mitochondrial membrane. Okay, this contains a proton channel. This contains a proton channel that is a C subunit. C subunit. This C subunit has proton units, proton pumping units. Okay, in C subunit the number can vary from 8 to 14. Some ATP synthesis will have 8 C subunits, some will have 9, some will have 10, some will have 14. These subunits transport protons back into matrix. Protons back into matrix. Okay, next it has A subunit and B subunit. Okay, A, C. C is divided into 8 to 14 parts, A and B. Okay, and this F1 has subunits called as alpha, beta, F1. Okay, alpha, beta, gamma, delta and epsilon. Okay, epsilon. These are the subunits of F1. In this, 3 alpha subunits will be there, 3 beta subunits, and in between these subunits, gamma subunit will be there. Okay, this gamma subunit is very important. Okay, this is gamma. This is gamma subunit. Okay, here we'll have alpha, beta, alpha, beta, alpha, beta, alpha, beta. Okay, 3, 3. Okay, 3 alpha, 3 beta. Between that, this gamma subunit will be there. In the center, gamma will be there. Okay, this F1 has catalytic activity. Catalytic activity for ATP synthesis. For ATP synthesis. So, in this F1, beta subunit, especially beta subunit, will participate in ATP synthesis. Okay, this 3 beta subunits are there, and these are the active sites. These are the active sites. These beta subunits will have active sites which help in ATP synthesis. Okay, and this ATP synthesis will occur in 3 sequential steps. Sequential steps. Okay, first what will happen? ADP and inorganic phosphate binding to the one beta subunit will occur, and second, ATP synthesis will occur. Next, ATP release will occur. Okay, and this beta subunit will be in 3 conformations, that is in 3 shapes. Okay, 3 conformations. What are those 3 conformations? Loose conformation, which will bind ADP and inorganic phosphate, and tight form, tight conformation, which will bind ATP tightly, and there is an open conformation where there will be a release of ATP. Release of ATP. Okay. Okay. So this will be like this. Three subunits. Okay, in the center we have gamma subunit. Okay, if this is in open form, this is in loose form, this is in tight form. Okay, here ADP plus PI will be there, and here in the loose conformation ADP plus PI will be there, and here ATP synthesis will occur. Okay, ADP plus PI. By flow of protons through C subunit of F0 will cause rotation of this gamma. So flow of protons through C subunit, C subunit will cause rotation of gamma subunit. Okay, gamma subunit. So how many degrees it will be rotated? 120 degrees rotation will be there. Okay, with this rotation, T form will be changed into O form, L form will be changed into T form, and O form will be changed into L form. Okay, there will be a conformational change. That means here in T form ATP is synthesized, but in this T from this T form ATP cannot be released. Only in open form ATP will be released. So this conformation has to be changed from T to O. This conformation will occur with the help of flow of protons through C subunit, which will cause rotation of gamma subunit, and this gamma subunit will compress the beta subunits and causes change in the conformation of beta subunits. Okay, from T to O, L to T, and O to L. That means tight to open form, L to loose to tight form, and open to L form. Okay, then what will happen from this conformation? Okay, then here in a tight form this will be there, ADP plus PI. Okay, then this is gamma subunit with 120 degrees rotation. Okay, 120 degrees rotation, and this O form is converted into L form. It has ADP plus PI, and O form. Here ATP is synthesized in the T form, so that ATP will stay like that, and this T conformation is changed to O conformation, which will release ATP. Okay. For 120 degrees rotation, 1 ATP is synthesized. Okay, 1 ATP is synthesized. Okay, so for 120 degrees means 1 ATP. If it rotates 360 degrees, what will happen? We'll get 3 ATPs. That means for one rotation we are getting 1 ATP. Another 2 more rotations, it will release 2 more ATP. Total how many ATPs for 3 rotations? You will get 3 ATPs. That means 360 degrees will cause release of 3 ATPs. This release of ATPs are occurring with the help of proton movements from C subunit of F0, which is causing rotation of gamma subunit, and this rotation of gamma subunit will cause conformational changes in the beta subunit, and ATP synthesis will occur in beta subunit, and there will be release of ATP from beta subunit, which will be in the open conformation. Okay, this is how ATP is synthesized and released from ATP synthase complex. Okay, suppose if the C subunit has 10 rings, okay, 10 parts. Okay, so each ATP generated requires how many protons? 10 by 3. Okay, for 360 degrees 3 ATPs are synthesized. So for 1 ATP formation, how many protons are required? 3.33. That means 10 rings will transport 10 protons. Okay, 10 rings will transport 10 protons, and for 360 degrees we are getting 3 ATPs. Okay, for 1 ATP you require 3.33 protons. 3.33 protons. The actual value will vary, and previously they said that NADH helps in synthesis of 3 ATPs, but now they are saying that 2.5 ATPs are synthesized from 2.5 ATPs are synthesized from 1 NADH, and from FADH 1.5 ATPs are synthesized. Previously it is 2, because the research is going on and they are finding new processes every time, and they have reduced the ATP formation to 2.5 through NADH, and through FADH they have reduced from 2 to 1.5 ATPs. This actual value of these protons for a single ATP will vary. Okay. Next, we will see overall flow of electrons and synthesis of ATP. Okay. This is complex 1. Okay. This is complex 3. This is complex 4. And this is complex 2. And this is complex 5. This ATP synthase is also called as complex 5. Okay. This is F0. Okay. This is mitochondrial membrane. Okay. Inner mitochondrial membrane. And this is matrix. And this is, you'll take it as a intermembrane space or a cytoplasm. Intermembrane space. Okay. Intermembrane space. This is complex 1, complex 3, complex 2, and complex 4. This is called as a complex 5. Okay. There is movement of protons in complex 1, that is 4 protons from matrix to intermembrane space. Complex 2 doesn't cause any movement of protons. Okay, no protons. And electrons are transported to Q part. Okay, electrons are transported from NADH to Q. Okay, from here FADH will transport to Q directly, and these electrons are passed to complex 3, and from complex 3 the electrons are passed to cytochrome C, and from complex 3 you will pass 4 protons. Okay, from cytochrome C electrons are passed to complex 4. Okay, in the previous lecture of part 1 of biological oxidation, we have seen in complex 4 cytochromes are utilized for formation of 2 water molecules. Okay, 4 electrons are utilized to form 2 water molecules, but the only NADH can transport 2 electrons. If you take 2 electrons, that means you will transfer only 2 H plus ions. For 4 electrons, you will have 4 H plus ions. For 2 electrons, you'll shift only 2 H plus ions. Okay, and half O2 plus 2 H plus will give rise to H2O. Okay, and here protons will be increased. And these protons will be transported through F0 complex and causes rotation. Okay, and they will help in synthesis of ATP. Okay, release of ATP from ATP synthase complex. Okay, this is about ATP synthesis through chemiosmotic hypothesis. Okay, next we'll see inhibitors of electron transport chain. Inhibitors of electron transport chain. Okay, complex 1 contains FMN FES. Okay, NADH will donate its electrons to complex 1. Next they will donate to Q and they are transported to complex 3, complex 3, complex 1, complex 3, and complex 2 will transport electrons to Q, and this complex will transport electrons to complex 4, complex 4. Okay, this complex 4 will have oxygen, this will have cytochrome C and C1, this will have FAD and FES. Okay, then what are the inhibitors of complex 1? Here the transport of electrons from complex 1 to Q is inhibited by inhibitors such as pyridine A, ammobarbital, barbiturates, okay, ammobarbital and rotenone. These are the inhibitors of complex 1, transport of electrons from complex 1 to Q. And here in complex 2, succinate will help in formation of fumarate. Okay, fumarate. This step is inhibited by malonate. This will prevent formation of FAD, that is one thing, and carboxyl will prevent transport of electrons from complex 2 to Q. Okay, carboxyl. Next, complex 3 is directly inhibited by the compounds such as British anti-lewisite and anti-mycin A. And complex 4, it contains oxygen. Those compounds which can avidly bind with the oxygen can inhibit complex 4. So which are those compounds? H2S, okay, H2S, carbon monoxide and cyanide. All these three compounds will bind with the oxygen and they will inhibit complex 4. So whenever these complexes are inhibited, then electron transport to the complex 5, that is ATP synthase, will be stopped and there will be no generation of ATP. Occurs. Next, we have inhibitors such as oligomycin. This will block completely oxidation and phosphorylation. How they will block? They will block flow of protons through ATP synthase. Okay, they will block flow of protons through ATP synthase. If that is blocked, more amount of protons are accumulated on the cytoplasmic side. Okay, and if more amount of protons are accumulated on the cytoplasmic side, then the electron transport chain can't work properly, so it will completely block oxidation and phosphorylation. Okay, next, attractyloside. This inhibits oxidative phosphorylation by inhibits transport of ADP, and that is ADP in and ATP out. It will block these steps: transport of ADP into the beta subunit and transport of ATP out of the beta subunit. That is out of the mitochondrion. Take it as an out of mitochondrion. Okay, this will inhibit transport of ADP in ADP. ATP out of mitochondria. Out of mitochondria. So this will inhibit transport of ADP and ATP out of mitochondria. These are the inhibitors of electron transport chain. Next, we'll see one important topic that is called as uncouplers. This is frequently asked in the exam. Okay, they will dissociate oxidation from phosphorylation. That means these compounds will cause only electron transport chain, but ATP is not synthesized. That means the flow of electrons will be there until complex 4, and from that, this after that complex 4 step, ATP synthesis will not occur. This complex 5 is separated from the complex 4. The most this uncouplers are divided into physiological and chemical uncouplers. This chemical uncoupler is 2,4-DNP. What this 2,4-DNP do? They will increase permeability of inner mitochondrial membrane, so that normally this inner mitochondrial membrane is not permeable to protons. It does not allow protons to flow back from intermembrane space or cytoplasm to matrix. But this 2,4-DNP, they will increase permeability of inner mitochondrial membrane to H plus ions or protons. This will increase. Okay, this increases the abnormal movement of protons from intermitochondrial space or cytoplasm to matrix. And physiological uncouplers are thermogenin, thyroxine, light amounts of bilirubin are helpful, or they act as a physiological uncouplers which helps in production of heat. production of heat, production of heat, which helps in maintenance of body temperature in a cold climates. Okay, this thermogenin is present in which tissue? Brown adipose tissue, brown adipose tissue. Okay, especially important for newborns because they'll enter into the external environment and they will feel very cold. So newborn babies will have high amount of brown adipose tissue, which will release a protein called as thermogenin, which uncouples the oxidative phosphorylation, phosphorylation. So this ends the biological oxidation.